Well, thank you so much for the opportunity to speak here today. And thanks so much, Angela, for a superb talk. I mean, you've really identified a number of the core themes with adaptive platforms. And I want to kind of build on that. I wonder if I could get my slides, or do I have my slides? Yes, we do. Okay. Um, so I think there are two quite separate concepts here that need to be kept in mind. One is the adaptive nature of trials, and the other is the concept of conducting trials on platforms. What I want to do is talk about one of these, which is an adaptive platform trial called REMAP-Cap, and just provide you a little bit of a background to it and some insights into what we've accomplished and really what we're learning. Because I suspect everyone here feels, I'm a little uncomfortable about this because this seems new to me and I don't know anything about it. You are not alone. Everybody who's doing these trials feels exactly the same way. They've been around for maybe 15 years, and that was in oncology and breast cancer where they were initially introduced. And so the power and the transformational power, I think, is something we're all really just beginning to understand. So welcome to the club. So from our perspective, pandemics and epidemics have been an inescapable part of our history as a species on this planet. These images show some of that toll going back as far as 5,000 years ago, where there's evidence of pandemics in China, to the Middle Ages with the bubonic plague, which fundamentally transformed the European genome. You can look at things like malaria. Malaria has transformed the genomes of people who live in tropical zones because it's beneficial if you have abnormalities in hemoglobin structure that protect against the pathogen. So we as a species are made up of all of the lessons of pandemics and infectious diseases that we've been exposed to over our lifetimes. Until the 21st century, the greatest of these was at least in the lifetimes of people who are around on the planet today, was the 1918-1919 influenza pandemic, which killed approximately 40 or 50 million people worldwide. But in the past, the main impact of a pandemic has been to select the survivors from the non-survivors. And by selecting the survivors from the non-survivors, it has changed the way we respond to future infections. That changed in a very profound way about 70 years ago. So during the 1918, 1919 pandemic, if you had severe influenza, there was nothing that could be done. There were no antibiotics to treat a superinfection. There were no mechanical ventilators if you developed respiratory failure. All that could happen was that you were cohorted in an area where somebody could provide you water, food, wipe your brow, and basically provide you with compassionate care until you either got better or you died. That changed, as I said, about 70 years ago. And again, it was a pandemic that changed this. It was the polio outbreak in Europe in the early 1950s. An anesthetist by the name of Bjorn Ibsen in Copenhagen got the bright idea when he was faced with children who were developing respiratory failure from the polio virus that maybe he could keep them alive by connecting them to external oxygen sources and having a group of medical students and residents bag them. So he recruited hundreds of medical students and residents, tracheostomized these children, and they would sit at the children's bedsides 24 hours a day, pushing on a bag and giving them oxygen. And that allowed them to survive. The mortality rate dropped from over 80% down to about 20 to 30%. Many of these children actually survived the pandemic. But what the other thing this did is it opened the door to a whole new specialty, and that's the specialty of intensive care, where rather than treating the disease, we support the patient while the disease is either being treated or much more frequently while the patient is getting better by themselves. And I don't think we realized how much the specialty of intensive care is grounded in public health and pandemics until the start of this century. Back in 2003, people in Toronto were very familiar with the SARS outbreak, where we were suddenly exposed to a lethal new virus, people who were otherwise healthy showing up with profound respiratory failure, and many of them ultimately dying. We wanted to study that. We weren't very successful. 2009, the H1N1 pandemic. We thought influenza was a sort of nuisance infection, and yet lo and behold, people were showing up in large numbers in intensive care units with influenza. And then, of course, most recently, the coronavirus pandemic. So as I said, we made efforts to try to study these diseases. There was, I think, the response, in a sense, as Angela says, the responsible clinician to something that they're unfamiliar with is, let's study it. How can I treat this patient better? How do I change what I'm doing so that patients have a better chance of surviving? But we were profoundly unsuccessful. During SARS, there are a few good cohort studies that describe the clinical course of the disease. During the H1N1 pandemic, again, large cohort studies, thousands of patients that describe the risk factors for severe H1N1 influenza, but only a handful of clinical trials, most of them small, underpowered, and by the time the studies that could have answered these questions got up and running, the pandemic was over. So there was a real disconnect here, I think, in the clinical research community, wanting to understand how to better treat these diseases and recognizing that they were going to be part of our future, but not having the tools to do so. So back in 2011, we had a meeting here in Toronto that brought together some international bodies of clinical researchers. INFACT is the International Forum for Acute Care Trialists. It's a network of investigator-led research groups in critical care. ISARC is the International Severe Acute Respiratory and Emerging Infections Consortium. It's a group of people who do public health research and emerging infectious disease research. So we got together to fundamentally address the question, how can we be better prepared for the next time? Because we didn't know what the next time was going to bring. We didn't know when it was going to happen, but we could be pretty sure that what we saw in 2003 and 2009 were not isolated incidents, and we would have to deal with this in the future. And so the one core notion that came out of that meeting was that if you're going to conduct research during a pandemic, the processes have to be in place prior to the pandemic. You have to have the trial up and running so that it can simply be shifted to study the pandemic rather than having to develop a protocol, go through all of the approval processes, get the funding for it. There's not enough time during a pandemic to do that. So the question was, how do you create this kind of model of a trial in waiting or a perpetual clinical trial? And what we did is we turned to a couple of people who were at that meeting, Don Berry and Scott Berry from Berry Consultants, Roger Lewis, who was one of the authors of this particular paper, who had been championing this concept of a platform trial. A platform trial was really a perpetual trial that could study multiple different interventions, could study them over time, and could adapt as it moved forward to respond to whatever came up in the future. So I want to just, Angela went over this very nicely, but I want to just follow it through a little bit further because I think understanding the subtleties and the differences between conventional trials and platform trials is helpful in understanding the potential power for a research platform. So as Angela said, a conventional trial focuses on an intervention. You take a population of patients, they have multiple different things that could alter the course of the disease, and those are distributed randomly throughout the population. What you then do is you randomize those patients to an intervention or not receive the intervention. All the other characteristics of the population are likely to be distributed equally amongst them. The only thing that's systematically different is the intervention. And therefore, you can draw inferences about whether there's a cause-effect relationship between the intervention and the outcome. That is the basis for scientific research, is having a control group and a study group and having randomization, the power of randomization, eliminate other potential confounding variables. But as Angela showed, it's really inefficient. You have to make a whole bunch of assumptions at the start of the trial. You study one thing and you come up with one answer. And often the answer is maybe more research is needed. Platform trials are different because they study a disease or a condition rather than the intervention. So rather than studying an antibiotic for pneumonia, you study pneumonia, and the antibiotic can be one of the things that you study, but you could study other interventions for pneumonia. So here is, for example, with REMAP-Cap, we can look at multiple different aspects of the support of a patient with community-acquired pneumonia. We can look at antibiotics, we can look at respiratory support, we can look at immune modulation, we can look at statins, we can look at anticoagulants or antiplatelet agents, we could look at music in the room. We could look at whatever you want that you thought might have an impact on outcome and look at it at the same time, because all of these are being studied on a common platform. And just some terminology here. We talk about things that are in a general therapeutic mode as a domain. So an antibiotic domain, you might be looking at piperacillin tazobactam, a third generation cephalosporin, and a carbapenem, and comparing the effectiveness of those. But you could have another domain that looked at ventilation or respiratory support, which could look at a mode of intervention of positive pressure ventilation versus high flow oxygen, for example. We separate these different domains out, and we are evaluating the effects of an intervention in a particular domain. So REMAP-Cap was the output of a lot of discussions about being better prepared for the next pandemic. And REMAP is an anagram. It stands for randomized, embedded, and that concept's come up. The idea here is that trials really should be embedded in the mechanisms through which we provide clinical care. It's multifactorial because we can study multiple different things at the same time. We use an adaptive model, statistical model. That's not necessary in a platform trial. You can run a frequentist platform trial as well as a adaptive platform trial. In fact, a factorial design is a kind of an adaptive frequentist trial. And it's a platform. So the idea here is that it is perpetual and that it's ongoing. Now, I want to just that's what a platform is. And I think the term platform trial is a wee bit of a misnomer because it's not, the emphasis is on the trial and that, not the platform. And it's a problem from a funder perspective because funders don't want to fund infrastructure. They want to fund research, and they call the research a trial. But to actually do a platform, you have to have a robust, ongoing platform to conduct the trial on top. So I think that it's useful to separate the concept of trial from the concept of platform. But I want to go into a little more detail into this concept of being adaptive as opposed to frequentist. So there are two different ways of looking at the world. There's the frequentist approach, which most of us were schooled in, and that you ask the probability of the data given the hypothesis. You state a hypothesis, and then you say, how likely is it that if I didn't assume this was right, I would have seen this? And that's what the p-value essentially tells you. Bayesian is a different. Bayesian analysis turns that around. And it says, what's the probability of the hypothesis given the data? And it recognizes that you're continuously gathering data. You had data before the trial, you're gathering during the trial, and you're updating the probability of the conclusions that you draw. It's much, much more like how we practice medicine. We're Bayesians because we say, well, there's this, 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 and this. There's not this. On balance, I think we're dealing with this, and so I'm going to do that. So here is how you interpret a frequentist p-value. It is the probability of observing a result that is as or more extreme than that you saw in the trial on the assumption that there's no effect. It's really tortured thinking. And it also emphasizes the idea that there is a yes-no answer. There isn't. This is a probability. We've arbitrarily set it at 1 in 20, less than 1 in 20. But it's, you know, if you have trouble getting your mind around adaptive trials, try thinking what a frequentist trial is, because an adaptive trial is very different. And it comes from the work of Thomas Bayes, who said, look, we all start off with ideas, with biases, with knowledge. These are our priors. And we update our priors as the data come in, and that's the process of learning. So in a Bayesian approach, you deal with what's called the posterior probability. After you've done the trial, what is the probability that the treatment is effective? And that's what we want to know. Again, it's not, is it effective or is it not effective? It is, what is the likelihood that what you have seen is effective? And to me, that's just really comforting as a clinician. I don't believe in absolutes. I do want to do on average what is better for my patients. So you've seen this, but it is important to just go through it again. This is the process for adapting in an adaptive trial. All of this is developed in advance, and that's critical. You're not making up the rules as you go. You're sitting down and you're saying, if this, then that, if this, then that. And you've got an algorithm that you test a priori by doing extensive modeling to find out what happens under those different assumptions. Then you set that algorithm loose on the data as it comes in. And as the data come in, the computer's kind of clunking through this, and it reaches an if this, then this, and it responds appropriately to that. And that may be that you've been a stopping rule, that you now can draw a conclusion that intervention X is harmful. You report that just as you would for a conventional clinical trial. It may be that we haven't reached a stopping rule. You continue the process of recruiting patients, analyzing the data. In REMAP-Cap, we don't have a set number of patients that we reanalyze after. It's actually driven more by time buckets than patients. But this goes on in perpetuity. And here's how it worked. This is a hypothetical situation, but a clinical trial where you start off with 3 different drugs in usual care. You recruit the first 300 patients, and after 300 patients, you see that usual care and drug B are doing a little bit better, and so you randomize more patients to this. This is called response adaptive randomization. So one of the algorithms in the trial is, let's give more patients the drug if the drug seems to be doing better. And so you do that. If you're wrong, it'll revert on the next analysis and you'll go back to more of a 50-50 thing. If you're right, it will continue to assign more patients to the arm that seems to be doing better. And here you see that by 700 patients, you're now down to 2 different arms, a conventional arm and drug B. And by 1,100 patients, you've reached the conclusion, you've crossed the threshold that drug B is superior. And in theory, this is knowledge translation, because you can say anybody in the trial is being randomized to this, and the randomization ratio is 100 to 1 or 100 to 0. Everybody gets that intervention. So just to get at that issue of how you ingrain information from platform trials into clinical practice. Obviously, if this is something novel, it's going to have to go through a regulatory agency. Okay, so REMAP-Cap is designed as a adaptive platform trial. It was set up to study community-acquired pneumonia because that's the phenotype of pandemic infections. That's what SARS looked like, that's what H1N1 looked like, and that's what COVID-19 looked like. But we had things at the outset that we thought might be beneficial for a severe respiratory infection, corticosteroids, maybe antiviral agents, maybe immunomodulating agents like interferon beta. There was some suggestion with SARS that might be beneficial. So we started with a few interventions that we studied. We grouped them into different domains, but we learned as the pandemic proceeded and as new data emerged, that there were other possibilities. There was a suggestion, for example, that vitamin C might be beneficial in severe respiratory infections. A lot of people were commenting on thrombosis as being a complication of COVID-19. So we added an anticoagulation arm and an antiplatelet arm. Angela talked about how SARS-CoV-2 interacts with the ACE receptor. Well, why not block the ACE receptor? So we added an arm where we looked at modulating the ACE axis. So we can add interventions as we learn more about the disease, and we can drop interventions as they reach a threshold. So I've gone through this. The terminology, the domain is a set of alternative and competing interventions. So in Angela's trial, hyperbaric oxygen would be probably a domain by itself, and the comparator would be no hyperbaric oxygen. The intervention is the options within that domain. And then you've got a regimen. And the question came up about randomizing to more than one domain. We do that all the time. We've randomized to as many as 7 different domains because you can actually look at the interaction between differing interventions when you do that. And that's focusing on the combination or the regimen. One of the things that allowed us to do is to look at what happens when you are on both steroids and IL-6 receptor antagonists. And in fact, you get a synergistic effect from those two different agents. So a domain conclusion, the domains are analyzed independently. You set in advance a stopping boundary, and once you reach that stopping boundary, you stop that domain and you report it, just as you would for a conventional clinical trial. And those results inform all the care of patients, hopefully, beyond that. And I think that has been the case in COVID-19. Most people were treated as results emerged from a number of platform trials. In REMAP-Cap, our criteria are superiority. It's an odds ratio, posterior probability of 99% that the odds ratio is greater than 1.2 for benefit. So a positive odds ratio suggests benefit. Equivalence, a 0.9 probability that the result is between 0.8 and 1.2. And inferiority, posterior probability of 99% that the odds ratio is less than 1. So that's the sort of statistics for this. Another beautiful feature of a platform trial is that the sites have autonomy. They can decide what domains they will participate in, and within those domains, what interventions they will evaluate. And so you're actually going to be studying those things that clinicians think are the most useful, potentially useful for treating the disease you're interested in. And I think it's something that our sites have been really quite fond of. A site that, for example, had strong views on steroids could say, we're not going to participate in that. A site that was doing a competing trial in respiratory support, we're not going to participate in that. But they could choose which domains, as long as they agreed that within a domain, they would choose at least 2 interventions, including usual care to randomize patients to. So, as I said, REMAP-Cap was set up to study community-acquired pneumonia, pragmatic inclusion criteria, and at least in its interpandemic phase, its primary outcome was all-cause mortality at day 90. It's a complex structure. There's multiple different working groups that oversee the different domains. There are regional committees. There's a data management committee. There's a data safety monitoring board. There are priority committees. And then there's an overall international trial steering committee, which is currently chaired by Srina Murthy, who's based in Vancouver. So REMAP-Cap is currently led by a Canadian. Move this forward. There we go. Okay, so that was REMAP-Cap up to January of 2020, when we began to get these reports about a strange viral disease that seemed to be breaking out in Wuhan, China. And all of our worlds changed at that time. The next steering committee meeting we had for REMAP-Cap, somebody made the comment, I don't think we're ready yet. If we only had another year, and we all said we don't, we're going to have to build this airplane as it's taking off from the ground. And so we very quickly expanded REMAP-Cap to increase the number of sites. At our peak, we were at about 365 sites around the world. In Canada, for example, we've recruited, I think, 1 patient by February of 2020 to the non-pandemic version of the trial. Many of our sites are concentrated in Europe, and I'll say a little bit about that because the UK is the dominant force there, and the UK has done some really innovative work in terms of thinking about clinical trials. But we are recruiting from every country, every continent now. This is old. We now have sites in South Africa. So we also, in the latest pandemic version, we were primarily focusing on critically ill patients. We expanded this to have 2 different states. Severe state was patients who were receiving organ support, and moderate state patients who were not receiving organ support. And this proved to be quite interesting in terms of the results that we got. Sorry, this is not moving well. Good. So we also changed our primary outcome for the pandemic phase. We wanted to be able to make decisions more quickly than having to wait 3 months for 90 days. And so what we did is change our primary outcome to organ support-free days over 21, an ordinal scale where if you died, you got a value of -1, and otherwise it's the number of days that you were alive and free of organ support. And this is shown on a heat map where blue is good, means you're free of organ support. Red means is bad, you're on organ support, or if it's dark red, that you're dead. And so you'll see this in some of the data coming from the trial so far, how that works. This is REMAP-Cap. This week, we have now recruited almost 13,000 patients worldwide, just over 10,000 with COVID-19. We have 61 different interventions in 17 different domains, 325 active sites currently around the world. And probably the most important thing is the number of differing comparisons, and it's about 22,000 right now for the trial. I want to just show you some of the data from this and to show you, because it also explains how a platform trial can work. We're using a Bayesian model. So in mid-June, the recovery trial in the United Kingdom reported that corticosteroids could reduce mortality by 12% in the sickest patients. We were studying steroids and we said, okay, we've now got external data. We're going to stop this even though we've not reached a stopping point. And so we analyzed our corticosteroid data in a much smaller group of patients, 379, but there was benefit, as you may be able to appreciate on the slide here. This is the control group or usual care group. Patients receiving steroids had less red and more blue. And so this was at about a 0.95 level, which to me, that probability of benefit is reasonably good. We published this in JAMA. So sorry. We then looked at another area we were looking at was interleukin-1 receptor antagonists, and we showed that either tocilizumab or sarilumab were superior to usual care. We published this in the New England Journal of Medicine in, I guess, February of 2021. We looked at anticoagulation. And this was really kind of intriguing because two things about this. Number one, we were using these two different states and we saw that there was a differential treatment response contingent on whether you were critically ill or not critically ill. If you were not critically ill, anticoagulation benefited you. If you were critically ill, it probably harmed you. So that was an important thing to disentangle a potential treatment effect. The other thing that I think was critical about these two papers is that they were a collaboration across platforms. 3 platforms, ATTACK, REMAP-Cap, and ACTIVE, the NIH trial in the United States, said, look, we'll just pool our data into a single trial. And that is the power of platforms, is that you can work across platforms and share data. It does mean rethinking how we think about research, but it is really powerful. We had over 3,000 patients treated with anticoagulants by the late fall of 2020, and we now had an answer to that question. We've looked at longer term outcomes. These are, I think, some really intriguing data. The two things that stand up as improving long-term survival in COVID-19 are IL-6 receptor antagonists and antiplatelet agents. So either P2Y12 inhibitors or aspirin result in an increased survival at 6 months. Didn't see it at 90 days, but at 6 months, that effect is there. We have, we've, I think, published about a dozen papers. We have 7 or 8 in JAMA, 4 in the New England Journal, and we've got 2 coming out this month, one on vitamin C, one on statins. Vitamin C, about 2,500 patients, and I can't give you the precise details because unfortunately the journals want them embargoed, but don't use vitamin C. Do think about using statins. Statins are this is a really interesting insight into the potential role for statins in infectious diseases. And that should be coming out hopefully a few weeks in the New England Journal of Medicine. So this is what we've learned in REMAP-Cap so far. At the start, we were getting pushback because people thought it was unethical not to give people hydroxychloroquine. It was, may have been unethical, but it was a smart idea because hydroxychloroquine increased the mortality rate or increased adverse outcomes in sick patients. Lopinavir, ritonavir, same thing. It was not beneficial. It was actually detrimental. And you put the two of them together and patients really did do significantly worse. It's why we do research. I mean, many people thought hydroxychloroquine was going to be the answer. It's not, and you need to stay away from it. Many people thought hydrocortisone was going to be a problem. Like, why would you give steroids to somebody with a viral infection? It'll go crazy. Well, no, it didn't. It actually improved outcomes. IL-6 receptor antagonists. I was incredibly skeptical that IL-6 receptor antagonists would have any value in COVID-19, and I was wrong. They do improve outcomes. I would have thought IL-1 receptor antagonist would work. Doesn't seem to in this particular population. Again, we saw anticoagulation, we've convalescent plasma where there was no significant effect, although a suggestion of an effect in immunocompromised patients, and we're studying those patients currently. Vitamin C, potential harm, ACE2 inhibitors, great biologic rationale, except it doesn't seem to help. It probably harms patients. So we've learned an awful lot about the management of COVID-19 through this single platform trial. We're recruiting from multiple sites around the world. These are the last few slides, and the data are out of date. We're now up over 1,000 patients in Canada. But I did want to point out two things. One is the UK, where the majority of patients are coming from. The UK made a decision in its healthcare system to embed research into the process of care and designated REMAP-Cap as the trial for patients who are critically ill. And so we're getting most of our patients from the UK. It was a brilliant idea, and it's something we need to be encouraging governments elsewhere to do. But the other thing that's interesting is we're also recruiting a number of patients from resource-limited areas. Nepal, India, and Pakistan have recruited well over 1,000 patients to the trial, and they're randomizing the patients and collecting data through a registry, a much more simple model, but they are not only recruiting and adding to our global knowledge, they're allowing us to look, is there geographic variability in treatment responsiveness? So we are recruiting from around the world. As a platform trial, we've got multiple partners and we've got multiple funders. And it's part of, I think, the critical factor. It's a critical factor in maintaining viability going forward. We just learned we've got more funding from Ireland yesterday, and so hopefully we'll be able to keep going. But the critical thing here is maintaining the platform more than it is doing any particular trial. And these are some of the particular funders who are involved, CIHRs, funding us in Canada, and multiple different agencies throughout the world are providing support for REMAP-Cap. So I wanted to end there and would be very happy to take any questions you have. Thanks.